So it's my great pleasure to introduce the Bill Paul lecture today. So for those of you who are maybe younger generation, Bill was a shining beacon for NIH community. He was at the NIH for 45 years, and 10 years after his passing, it's great to still be honoring him and his contribution. So Bill was trained at NYU with Badouche Benasseraf, moved in the 70s here to start his own lab, and he was heading the laboratory of immunology for many years. And of course, he's remembered for many seminal contributions in the field of immunology, especially the discovery of IL-4, all the delineation of Th2 immune responses, the interplay between T cells, B cells, mast cells, basophils, you name it, the whole shebang of immunology, of course. And I was very struck when looking at Bill's extensive list of publications to see the first paper when they delineate the discover IL-4 and report on the discovery. That was in 1982. And the title was Identification of the T-cell-derived B-cell growth factors distinct from IL-2, right? So at that time, we didn't call it IL-4 yet, but no, it was clearly something very seminal. Of course, Bill also created the Bible of immunology, fundamental immunology, which is this gigantic book we all have on our desk, we refer to. It was started, the first edition was in 1984. And in one of the editions, he reports that at that time, he wanted to really acknowledge the explosion of the field of immunology and try to consolidate the whole knowledge in the field. And he felt in 1984 that 800 pages would be enough, right? So move forward, 2022, Martin Flajnik, Neville Singh, and Steve Holland have released the 8th edition of Fundamental Immunology, and now we are clocking at 1800. So I think we are still in this kind of explosive growth of the field, and we still need to consolidate, and it's great that the name of Bill is still on this Bible of the field. And Bill also represented the best of NIH because he moved from very fundamental immunology, basic immunology, to the more clinical, and of course he managed the Office of AIDS Research in the 90s at a crucial time when a lot of therapies were being developed. He also founded the Vaccine Research Center, so these are massive contribution for the field of public health. Bill was a mentor to many great scientists. I'm just going to name a few just to inspire people. So Maureen Howard, who was the first author on the IL-4 paper, Clifford Snapper, Melissa Brown, Bob Sider, Mark Davis, Barbara Fazekas de Saint-Grove, Tomohiro Yoshimoto, and Jeff Zhu, of course, and many others. At the more personal level, so I was not a mentee of Bill per se, but I joined Ron's lab in 2000, Ron Germain's lab in 2000, and he placed me at that desk. And literally, my desk was across the door from Bill's office, so I would bump into him all the time. And I would really benefit from his wisdom and his kindness and his ability to discuss. So I'm a theoretical physicist by training, and Bill always listened to my crazy ideas and what we are trying to push forward with Ron, and with a twinkle in his eyes, and asking probing questions in a very kind way, but kind of telling me, calm down, try to look at this, and so on and so forth. And especially at that time, we had this competition for model of tuning. It was a lot of fun discussion. And Bill kept helping me even when I moved to Sloan Kettering and pushed me to do more inflammatory work, so it was great. And just for, again, the young generation, like the LI, the Laboratory of Immunology at that time, we had this seminar series, and giving a seminar at the LI, you had Ron and Bill in the first row, and it was, a bit intimidating. So to me, it reminds me of an episode of Law and Order where you have a good cop and a bad cop. I'll let you guess who the bad cop is, Ron. Anyway, for Bill's memory, it obviously shines over all of us. I mean, we hope that we are following in his footsteps and fostering great science, and that's what we're going to do today. So I'm going to move to introduce Ruslan Medzitov, who is our speaker for today. We greatly appreciate that you accepted the invitation to deliver this lecture. And every year when the Immunology Interest Group start nominating people for the WALS or for the Bill Paul lecture, Ruslan's name come about, but there are rules. We cannot invite Ruslan every month, so we have to wait a little bit. So I think you delivered one of his lectures a few years ago, so welcome back. So to introduce Ruslan, he comes from a non-immunology background, which is, of course, a good thing. So he trained in biochemistry in Moscow, and then some evolutionary biology. I'm picking up from your Wikipedia page, sorry about that. And then most importantly, Ruslan moved to Yale, where he has been at since 1994, specifically in the lab of Charlie Janeway's. And then no pressure on our trainees, but like I think Ruslan's first of a paper, first contribution was literally a bombshell for the field because Ruslan and Charlie and co-authors reported the cloning of a human homologue of a TLR, the fly receptors, which they identify as the key receptors to build the innate response. So again, it's in 1994. I mean, there had been some statement from Charlie Janeway about the dirty little secret of immunologists that we needed some goo, we need something to really activate the immune system, and this paper was absolutely a game changer, which completely revolutionized the field. It created the field of innate immunology, and the rest is history. So again, no pressure on our trainees, but try to follow up on that. Ruslan, of course, didn't stop there. He went on to make amazing discovery over the years, from the field of inflammation, antigen processing, the way innate system speak with the adaptive immune response. And the silver lining of all of Ruslan's paper from a personal level is that sometimes it's a bit light on data, which is again great, but the concept is very clear and it's very thought challenging, and it's better to make a point with fewer data. It clearly shows that the idea is very, very strong. So again, the ratio of conceptual ideas to data is in the right place, not in the wrong place. I'm going to point out one of my favorite papers, which I would refer to. It's from 2012. It's titled, it's more of a think piece. It's titled Disease Tolerance as a Defense Strategy. And I think here we have a roadmap for many years to really try to understand how tissue homeostasis and how it relates to immunology and to response to infections comes about. So again, a very broad vista. I think it has helped a lot of people in the field. And I think it's a very good settings to have Ruslan deliver the Bill Paul lecture today, because it's really this global view of immunology, which is really great. So some internet thing. This is a CMA lectures. So there's a code there on the screen, 57689. And for people online, if you want to ask questions, please do type your questions, the live feedback, and we'll pipe in the questions at the end of the seminars. So again, it's great to have Ruslan here, and the floor is yours.
Well, thanks very much, Gregory, for a very kind introduction, and thank you for inviting me. It's a great honor to give Bill Paul's lecture. I have very similar kind of memories of Bill Paul being this nice, good cop and kind, and with a lot of wisdom, and he was really a monumental figure in immunology. And I have to explain the title, which is very schizophrenic. And the reason for that is that when I was asked to give a title, as it happens, you forget to respond. Then there is the email, and we need it today. I panicked and sent the emails from First Principles. I don't know why, it just popped into my... I had some talk related to that. But then when I was asked to send abstract and whatever, learning objectives or something, I forgot what I sent, and I thought, well, it's Bill Paul lecture, I should talk about allergy, because that was his field, and so I sent the abstract about allergy. And then this morning I was trying to figure out how do I get out of it, and I can't, so it will be about allergy, but I will, it's not going to be the talk about the immunology for first principles, which is, it's a different subject, but I will try to connected to some couple of points that are more general. So sorry for that type of title. That's a little bit misleading. So I just put them both here. That makes no sense now. So anyway, so I'll talk about allergic diseases, and I will talk about a couple of perspectives on it. The main point would be about why, what the question on the mind of many people who have allergies: why do we have allergies? Why do we have to suffer from it? And why is it becoming so common? And so it has to do with this trade-offs we deal with any type of defense systems. So just to put it a little bit of introductory perspective, so this is how we can think of different types of inflammation. and their intended causes and unintended sort of consequences, pathological consequences. We now appreciate that there are several different types of inflammation. So for now, I think we can distinguish 4 of them. The best studied is infection-induced inflammation. There is a clear intended purpose to protect from pathogens. Exaggerated responses lead to collateral damage and sepsis. With tissue damage, intended responses, tissue repair. When it's exaggerated, can lead to fibrosis, and solid cancer, solid tumors can promote solid tumor growth. And then the infection and damage is not the only problems in the world of animals. So noxious substances, toxins, noxious chemicals coming with food from usually plant secondary metabolites, as well as venoms. These are also very common. We don't appreciate them as much now because we are not generally exposed to them in industrialized world, but it played an important role in evolution. And I think that the defense from them is, if it's metabolic defense, detoxification, otherwise it's expulsion, neutralization, and so on. And that's the part that I think leads to, when exaggerated, leads to what we call allergies. So the normal counterpart of allergies, I think, is that defense from noxious environmental substances. And finally, the 4th one is it doesn't have any intended purpose because it's a consequence of mismatch between our evolutionary history and modern environment. And modern lifestyle, as we know, can be great, but it can have costs. And these are various modern diseases of metabolic, cardiovascular, aging-associated diseases. But the point here is that for allergies in particular, we can draw the same parallels that what we know in the more simpler, better understood case of infections, that you have immune protection, but it comes with a cost. And if it's exaggerated, then we call it disease. And more generally, if we think about the costs of defenses, which are historically always first were thought of as diseases, but we need to distinguish 2 very different types of costs of defenses. And this is true for any defenses, and it's very much true for inflammatory defenses. So there are 2 types of costs of inflammation. One is, on the right side, that's the more familiar one. It's unintentional but unavoidable loss of function, and at the extreme would be collateral damage. So that's just something we can't avoid, but it's part of the cost we pay for having defense. The other one is more interesting, and that is intentional suppression of lower priority functions. That needs to happen in order for the immunoinflammatory response to do its job. And that is more by design. But of course, if it comes with a cost because you suppress some other functions. And when it's exaggerated, it also can become problematic and can lead to disease. And both of these costs contribute to ultimate sort of costs of the inflammatory response and can contribute to inflammatory diseases, but these are very different types of costs. One is intended and well orchestrated, and it just can become exaggerated, and the other is just collateral damage, unavoidable, unintended consequence. Both contribute to the cost of on our fitness, and we can think more generally about these types of traits like defenses that are very important for survival and coming with the cost. Like any traits, in order to be evolutionarily stable, the benefit has to be higher than the cost. And as we can see here, anything in this green part of the square where benefit higher than the cost would be evolutionarily selected for. Anything in the red side would be selected against where it costs more than benefit. But in this area, in the where benefit higher than the cost, you can appreciate that if benefit is very high, acceptable cost can be very high as well. And this is evolutionary sort of point of view, and a patient or doctor point of view of the same exact plot would be like this. So anything on the right side of the square would be considered to be a disease, even though it's evolutionarily stable. And these are the conditions we experience, for example, in acute illness, when there is a very severe consequences of high magnitude defense responses, like inflammatory response or anaphylactic response. And the reason they exist is not because they're good for that particular person in that particular situation, but because on average, in the course of evolution, in very different environments, on average, that provided better benefit than not having that type of response. And incidentally, what's interesting is most of the studies of various types of experimental models of diseases, we draw the line somewhere where we just say, well, animal is dead or not dead. But in this way, there's a lot of very interesting biology that is hidden that lives over here. And this is because we don't know that biology well. That's why critical care medicine is also not where other branches of medicine are in terms of understanding how to help patients, because underlying biology is not well studied. And that's one area we're interested in. But in the context of this talk, I'm bringing it up because in the severe allergic reactions, not only can be a nuisance, they can be life-threatening. Anaphylactic reactions can be life-threatening. And the question is, what types of mechanisms enable it, and what can we do about it? So, to quickly remind you, what I will talk about is allergic immunity, or type 2 immunity for simplicity. This is to remind you that we have two types of immune responses to microbial pathogens. It's type 1 response, and to macroparasites as well as to allergens, which is an ill-defined category of stimuli, where we have type 2 response. In both cases, exaggerated response can lead to problems like sepsis and anaphylaxis. And also to remind you that would be relevant is that for type 1 immunity, we know basic principles, that there is combination of antigen with some immunogenic stimulus, typically something derived from conserved microbial structures detected by innate pattern recognition receptors. That's what drives type 1 response. In the case of type 2 response, we don't have good understanding of what the adjuvant effect is going to be. And it's not structural recognition. It's in some cases, some functional recognition, like enzymatic activities of venoms and other types of enzymes, proteases, lipases, and so forth. And sometimes it's other features that are still ill-defined. That makes this area still not well developed, not nearly as well as understanding of initiation type 1 immunity. It's an ongoing sort of question in the field. But generally, the idea would be it's some kind of a noxious activity when combined with antigen is what drives type 2 response. And the point of it is that the subsequent execution of this response will be protective. And that's what brings me to this point that when we think of allergies, we typically think of it as a disease state. But I think we need to distinguish between allergic diseases and allergic defenses. So what's the normal counterpart of allergic responses? For a while, the dominating thought was that, well, allergy is just a mistake the immune system makes, and then the normal counterpart defense from parasites. I think that's not entirely correct. I think the normal counterpart is defense from noxious substances, and there are many reasons to think that the response, allergic response, is not just a mistake. There is one nuance that I will explain in the end of the talk. It's not just a mistake because, as we know, like in the case of food allergies in particular, people who are allergic to certain foods, they are responding to the same allergen from the same food sources. So if it's a mistake of the immune system, it's making the same exact mistake again and again. So it cannot be just random mistake. And all allergy symptoms, of course, they all have to do with expelling something noxious from the body. And moreover, exact same reactions occur in people without allergies. If you just inhale dust particles, you'll sneeze and cough. If you touch irritants, you'll cause itching. If you have food poisoning, you'll have vomiting or diarrhea. So these are just generic responses against something that's noxious, promoting their elimination from the body. Allergic immunity is superimposed on those defenses, and those defenses are mostly operated by neuronal reflexes. And allergic defenses are superimposed on top of them, making them much more sensitive, antigen-specific, and much more problematic, potentially. So from that perspective, we can think of allergic defenses as the optimal execution of type 2 response. When it's exaggerated, it results in allergic diseases. And then we can, based on that, also predict that there is another area of pathology where these defenses are insufficient. So this is very much parallels what we know about immunity to microbes. You can have optimal immunity, you can have immunopathology, you can have immunodeficiency. In the case of allergic defenses, we don't know what in that left corner is with question mark, but this would be just a prediction that there are maybe a class of conditions which may not be, probably not known to be caused by that, but would be predicted to be caused by insufficient allergic defenses against noxious substances. So the question then we were interested in, how responses are initiated, but what I will focus on now is the one big enigma in the field is what makes somebody allergic or hypersensitive. Everybody has IgE responses, everybody has Th2 cells. They are triggered by appropriate stimuli, but only a subset of people become hypersensitive or allergic, what we would call allergic. And this is not all or none type of distinction. People can be allergic to different degrees. But what makes somebody allergic and hypersensitive, that's a big question. We don't know even how to define that state. What defines that state? We know IgE antibodies of high affinity are required, but beyond that, it's really not clear what exactly defines hypersensitive state. And so that's probably one of the biggest questions in the field, because we would want to be ideally able to revert that state. But the question that I will focus on in the first part, so my talk would have two parts. They deal with two aspects of the environment that, from very different perspectives, that make allergy problematic in modern world. The first one, is why is the incidence of allergic diseases increasing? So over the past few decades, increased dramatically. So the only way to explain it is through environmental influences. It cannot be genetic on that type of scale. And we're interested in what is it in the environment. It's a very hard question because so many things changed in the environment in the last 30 years, right? So it's easy to find something that has a very perfect correlation. And there are some usual suspects, like cleaner world, more sort of changed diets and changed microbiota and whatever anyone's favorite environmental factor is. And they probably all contribute to various degrees. And this is just a picture showing how environment contributes to incidence of allergies and how it very much relates to industrialization, eating foods that are highly processed, living in a clean environment, use of antibiotics, hygiene products, and so forth. And so clearly something about the environment, and it's been the long-standing sort of hypothesis that clean environments are the ones that promote development of allergies. And why that is that's the question that is we've been interested in. So the basic premise would be that the immune system evolved under conditions that are environmental conditions that are very different from what we enjoy currently. It's normal meaning evolutionarily normal. For us, it would not be desirable. But normal conditions would be very high microbial exposure and 100% parasitic infections. Every animal in the wild has parasites. And that would be the normal state. But the reason it's important is because that's what the immune system normally deals with. And that's what it evolved to deal with, to operate under those conditions. And in the modern environment, those conditions change dramatically and characterized by reduced exposure to microbes, parasites, and foreign antigens in general. So we wanted to ask to what degree that contributes to predisposition to allergies, how environmental factors influence allergic sensitization. And to address that, in that first part of the talk, what we did, we used so-called pet shop mice, which is mice you buy in a pet shop where they're bred not as pets, by the way, as food for snakes, but they're called pet shop mice. That's a better name. These are basically wild mice that are caught somewhere in the barn, and they are genetically diverse. They are very dirty. They have all kinds of microbes. And the goal was to investigate their immune system as compared to SPF mice, specific pathogen-free, that lives in vivarium and so on. And David Mazepus described the model with pet shop mice a few years ago and characterized some basic immunological characteristics of these mice and why they could be a good model for various questions. So this is what these guys look like. On the right, it's bulb C mice, clean SPF mice. On the left, pet shop mice. You see that they clearly genetically very diverse. They look very different. They behave differently. And they are full of this stuff on the right. This is not even complete list, but they got it all. They got protozoal, they have metazoan parasites, they have bacteria, fungi, viruses of all kinds. They are full of that. And, you give any one of these to SPF mice, you see that you infected them and you see whether they die. These guys have it all, and that's their normal state. That's one kind of actually pretty amazing fact, right? They have all this colonization. You can't even call it infection because they are not sick. It's just their normal state. And that should tell us something about what the normal state of the immune system, of the organism immune system have to deal with. So anyway, so we use this mice. So what this allows to do is that we are looking at genetically very heterogeneous group of mice that are full of normal sort of exposure to microbes of all kinds. And we're comparing it to what we normally use in the labs, SPF condition, inbred mice of a particular strain. And when we subject them to exact same protocol of allergic sensitization, this is the model where we use alum with subcutaneous immunization with ovalbumin. And that is a very robust model. In bulb C mice, they develop allergy. And then you can see here, if you challenge them, this is temperature drop. So this is the result of anaphylactic reaction. If it's severe, it will kill mice. And you can see that pet shop mice are really protected from it. This type of graph I'll show several times. This is delta in temperature. So the higher the graph, the stronger anaphylactic reaction. So as you can see again, these mice are protected, even though they make antigen-specific IgE. So that's the part that I think is really important, that they do make IgE just fine, antigen-specific IgE, but it doesn't result in anaphylaxis. And so then we asked whether, if we take our clean mice that are these inbred bulb C mice, to rule out that this is just something like really weird random genetic effect. And we cross-foster them with pet shop mice. So this is anaphylactic reaction in control SPF mice. If they're co-housed with dirty mice, they acquire some of the microbes, they become more protected. If they're fostered by pet shop mice, they are completely protected. So just acquiring these microbes from these dirty mice makes them completely protected. And that's consistent with epidemiological studies in humans living in a more natural sort of dirty environments is protective, growing up on the farm is protective, and so forth. And they're protected not because there's any kind of defect in the allergic inflammation, because if we bypass the sensitization state and just give them passive systemic anaphylaxis on the left, they would anaphylax just as SPF mice. And if we bypass even antibody part and just give them either a compound that induces mast cell degranulation in IgE-independent manner or induced histamine, they would anaphylax as well. And in terms of the antibody response, again, these are very heterogeneous genetically mice, but there is this common trend. So their T-dependent antibodies are elevated compared to SPF mice, as shown here with IgGs and IgE and IgA. T-independent antibodies seem to be there's the opposite trend, but that's probably the consequence of them mostly switching to T-dependent ones. So they have high antibody levels, and then what we did, we took about 20 plus very different, genetically distant mouse strains from Jackson Lab. We chose them based on their being maximally different genetically, and they're shown here, and we compared sensitization in all of them to pet shop mice, which are in the red. And as you can see, the responses vary, so this is for genetic reasons, because these are all SPF, but different genetic backgrounds. And we're comparing to genetically heterogeneous pet shop mice, which are red. And you can see they have this is again the same type of immunization, OVA and alum. And you can see they develop very robust IgG responses to OVA and alum, which in SPF mice, if you look at IgG2 in particular, it's almost not there. But pet shop mice develop very strong IgG2 response. And this, by the way, is reminiscent of what happens in humans, because alum is one of the adjuvants used in humans. And in humans, it can induce appropriate response to for vaccination against microbial antigens, so it will induce IgG2, for example. And that's not the case in SPF mice, but it is the case in these more naturally exposed pet shop mice. So they have elevated antibodies and protect from allergy. They generate both IgE and IgG in response to allergic sensitization. So I emphasize allergic sensitization, meaning use of alum. And that is unlike SPF mice, but similar to adult humans. So in adult humans, it will induce also both type 1 and type 2 response. And some of you may remember when Th1, Th2 subsets were discovered for a long time in the human field, it was argued that they don't have that clear distinction between Th1 and Th2. And that's generally true for adult humans. And so I'm not showing data, but allergen-specific IgG antibodies are protective from allergy. That's also known from various studies. And the question is, why is the antibody response to the same immunization different in pet shop mice and SPF mice? And that's where we found one, we got first surprise is when we looked at anti-over response, of albumin-specific response, in SPF and pet shop mice without immunization. So neither of those mice saw of albumin. They were not exposed to it. It wasn't in food. It wasn't immunized. And in SPF, as you would expect, there is no response. But in pet shop mice, there is a response. It's not a very high titer, but it's there. And if we take the SPF mice, if we take pet shop mice, put them in SPF conditions, so now we do cross-fostering in the other direction, then they lose it. So they grow up in clean environment, then they don't have that antibody response, reactive to ovalbumin, right? So they have to be dirty in order to have it, even though they didn't see ovalbumin. And then when we looked at so in order to characterize that further, we developed this antibody screening platform that allows us to look at specific epitope responses. These are linear epitopes, but nevertheless, that was very informative. So we use this system where it's a bacterial display essentially of different epitopes. And then this is showing that it works, that when we induce expression of the antigen, and then antibodies will recognize the appropriate epitope, and then if we use monoclonal antibodies as a control, it shows exactly in which position those epitopes are. And this is, on this graph, the amino acid position is on x-axis for ovalbumin. And these are two epitopes recognized by these two monoclonal antibodies. This is just to show that the system works. Then we use that system and compared antibody reactivities towards different epitopes from pet shop mice versus SPF mice, without and with immunization with ovalbumin. And this is what happens. So the top is SPF mice. So as expected, without immunization, there is no antibodies reactive to OVA. After immunization, you start seeing this various antibody reacting to different epitopes. But in pet shop mice, even without immunization, there is already plenty of reactivities towards epitopes of albumin. With immunization, it changes, shifts, and so the point is that even without immunization, there's plenty of the responses. The same is true with T cells. So if you take T cells from SPF, unimmunized, there is no response to either ovalbumin or KLH. When they're immunized, you start seeing a response to KLH, but not ovalbumin. Pet shop mice, without immunization, there's plenty of response to both, even though they haven't seen either one. And this is all quantified here. So this is reminiscent of what happens, what's been reported in humans. For example, in Mark Davis' lab, and others demonstrated that when they looked for specific antiviral epitopes responses in T cells, they see that they have T cells reactive to particular epitopes, even though, for example, the HIV epitope, even though they were not infected with HIV or vaccinated. And so that seems to be the general rule, and it's only not the case in SPF mice where you don't have that. So in that sense, to say that mice are, Mark argued mice are not humans based on that and many other things, but SPF mice are less like humans, dirty mice are more like them. And so this is just to show that there is this reactivity of T cells and bottom CD4 T cells. With Th2 responses, the situation is different, that there's not much of a type 2 memory recall response, but it's very strong with type 1 response. Okay, so conclusion from that part is that pet shop mice have pre-existing antibody responses, both IgG and T cells, and this must be specific to something, because they haven't seen those particular antigens, they must be cross-reactive to something they have been exposed to. And that this cross-reactivity is what allows what we detect when we do these experiments. And they generate this mixed Th1, Th2 response, which is, again, unlike SPF mice, but similar to adult humans. And so their immune system in a quite different state. So schematically, we can look at it this way, that in a true naive state, which would be mimicked by SPF mice, where in gray, naive T cells, and then you perform allergic sensitization, you generate Th2 and Ig responses, and they become allergic. They now will anaphylax in response to challenge. If you do oral tolerance, you expand Tregs specific, and that will be protected. But in the wild case, in the normal sort of natural case, there is already pre-existing natural sort of type 1 responses, and that protects them from developing allergy, even though they would have type 2 response. It's just they will not be hypersensitive. And that's what natural state is. Again, everyone has Ig responses, but not everyone hypersensitive. And the prediction from that is that pet shop mice should be susceptible to developing allergy if they are sensitized before they develop that state of the immune system, before they develop that antigenic experience where they now cross-react to all sorts of antigens. And SPF mice, conversely, should be protected by prior cross-reactive exposures. And so this is the experiment about the first part, where we took pet shop mice. So if you sensitize them as adults, they are protected. That's shown in green. If you and then we sensitize them at postnatal day 7, before they developed all this state of the immune system, exposed to all these allergens. And then at days 58, 65, we challenge them, and now they are allergic and they anaphylax. So if they're exposed to the given allergen prior to developing all this antigenic experience, then they're sensitive. That's the window of vulnerability, and that, of course, also fits with what we know in humans, where babies are much more sensitive to developing allergic reactivity. Okay, so then, yes, so, sorry. So then we to look at it in a different way, we took ovalbumin from different avian species and reptilian species, and to see whether there would be cross-reactivity if we can mimic that cross-reactivity in SPF mice. So we, again, sensitize them with alum-ova, but then we challenge them with either chicken ovalbumin or duck or macaw or even turtle. And these all have different distances in terms of amino acid sequences. And you can see that even as you go down in distance from left to right, reactivity goes down, but the reactivity is there even to these cross-reactive non-identical antigens. And more clearly, this is seen on this with our antigen screening system, where chicken ova is the one that is 100% match, and then it progressively goes down, but you still see these reactivities. So this is just to say that there is this cross-reactive immunity that can be even artificially developed here with just using one antigen. So this leads us to the model where we think of one aspect of vulnerability to allergies associated with modern clean environment is the following, that in the neonatal state, the only antigens the animal would see is self-antigens. If they develop in clean environment, like SPF conditions, then they also would be exposed to microbial, commensal antigens, and dietary antigens. But that doesn't fill out the entire antigenic space. There are many other antigens that are not exposed to. In the pet shop mice and also in natural conditions for humans, when you go from this neonatal state and you get exposed to this hugely diverse antigenic environment, you develop responses that through cross-reactivity will cover the entire antigenic space. And that's what makes them protected from developing allergic reactivity after that state is established. Because over there on the right, there would be gaps in what's covered by antigenic space, and if the allergen comes and it fits into that gap, which would be usually the case because the gaps are huge, then you develop allergies. In this case, you are protected. And in terms of the life history course, here the pet shop mice at the top would show situations similar to humans in natural environments, where this window of susceptibility is short. After, in the early stages of life, there's a short window before you're exposed to all the allergens. And that's when you're vulnerable. If you're exposed to allergens at that state, you can develop allergies. But then it quickly closes down by developing all these other reactivities and filling up antigenic space by various antibody and T-cell reactivities. In SPF mice, it's not cleared as much, and they can maintain the susceptibility to allergies. Humans in industrialized environment, somewhat similar to SPF mice, also that window is extended and they become more prone to developing allergies. So that's the aspect of the environment that we think has to do with the ability of the immune system to cover as much of the antigenic space as possible due to natural exposure to antigenic environment, and that's what provides protection. That's one aspect of providing protection. The second part that I will quickly go through has to do with another aspect of the environmental factor that makes allergies problematic. It's not about developing them or not, but what makes them problematic. And that has to do with, basically it can be described as a phenomenon of evolutionary trap. So evolution trap is when animals or humans evolve under certain conditions, and then in an environment change, they still react the same way, and that leads to their demise or disease, right? For example, turtles trying to cross the road, you turn them around, they still turn and cross the road because they didn't evolve with cars running around, this sort of thing. So in the context of allergies, what that means is that if it is indeed the way we think about it, is that it's a protective response against noxious substances, then once you're exposed to something that causes allergy, let's say house dust mite, in the wild, it won't be house dust mite, but it would be something similar. Let's say some pollen under some tree, from some tree. So basically all you do is don't sleep under that tree. Just go at the side of the river and you're fine. If some plant made your stomach upset, just don't eat that plant. Just remember what that plant was, this sort of thing, and then it's not a problem. Just like with all the other things in the environment that are harmful, we just avoid them. In modern environment, it's not possible. If you're allergic to house dust mite, the only way to sort of mimic what nature intended is not to live indoors, which is not an option. Right? So that's sort of that's the sort of principal evolutionary trap. And with allergies, we think, and there's plenty of evidence also with humans, that one important aspect of allergic defense is behavioral defense. It's avoiding exposure to something. Once the system detected that something is problematic, it tries to remember it and tries to avoid it in the future. So that would be behavioral defense. And that's what we studied. And I'll go quickly through this part, but I want to get to the final points. But the history of that is actually, there's been studies about 30 years ago in Brazil from Nels Voss group, where they looked at the question whether allergies would promote aversive behaviors. So here they made mice allergic to corn or peanuts, and then gave them choice to eat both, and then they choose one or the other. So this was over 30 years ago, so now we wanted to study that with a modern approaches to look at it more in more detail. And we use this what's called two-bottle preference test. We either make mice allergic or not, and then we give them a choice to drink clean water, regular water, or water of albumin, which is the allergen they're sensitive to. And it so happens that, which was good for experimental purposes, that they actually, if they're not allergic, they prefer the water with of albumin for some reason, but that was good for us. It made the result more clear. So in control mice, you can see they prefer of albumin water. If they're made allergic, they lose that preference, and then over time, they start avoiding it. And this is now looking at 48 weeks past. They now prefer drinking clean water and avoid of albumin water. And this response is antigen-specific because if we use BSA, the bottom part, then they're fine. They will continue drinking it. It's antigen-specific response, and it's a behavioral response. And the neurobiological aspects of avoidance behavior, there are some things known about them, mostly in the context of sickness. And there are these circuits that are involved in inducing, for example, anorexia and avoiding food consumption. It includes information collected through nucleus of salivary tract that comes from visceral sensory neurons, and then it communicates with parabronchial nucleus, which is a major brake on behaviors like food consumption. And that information also then passed on to amygdala to generate memories about these negative experiences and to avoid it in the future. And what we found is that if we make mice allergic and then expose them to allergen, all these areas become activated. These are areas that are activated when mice experience negative stimuli that induce avoidance behavior. So they all become activated. And that response in the brain is dependent on, and the behavior is dependent on Ig antibodies. So without Ig antibodies, avoidance goes away, and activation of these brain areas goes away. So it's an antigen-specific behavioral response. And because this Ig, it also depends on mast cells, not surprisingly, it's shown here. So then the question is, how do mast cells communicate with the nervous system, central nervous system, to promote aversive behaviors? And what we found is that there are two mechanisms, fast and slow. It's very much similar to some other types of aversive behaviors. For example, pain. There's a fast pain, which is mediated by A-beta fibers. This is very fast response when you withdraw from touching something painful. And there is slow pain mediated by C fibers, which is much more prolonged. So we think there's something similar, although mechanistically it's not through different types of fibers, but it's through different types of signals. So the first thing we found is that there is this hormone called GDF15, which is an extremely interesting hormone that is induced whenever there is something off. It could be pathogens, allergens, toxins, DNA damage, and so forth. And it induces these protective responses like nausea, anorexia, and at the extreme can be also problematic in cachexia. The receptor for this GDF15 is expressed in brain area called area postrema, which is responsible for vomiting, nausea, and these types of protective responses from toxins. And so what we found is that GDF15 is very robustly induced when sensitized mice are exposed to allergen. So exposure to allergen induces this very strong peak in GDF15 production. And this is just, I think, a time course there. And this production is dependent on IG and mast cells. And GDF15 is known to act on area postrema and to induce nausea and aversion. So that would turn out to be one mechanism, because when we block GDF15, this avoidance behavior goes away, and now they keep consuming of albumin that they're allergic to. The question is, how do mast cells turn on GDF15 production, because it was not coming from mast cells. And it turned out that it was coming from, it required a different pathway that operates in mast cell, which is leukotriene synthesis pathway. So these leukotrienes, and this is some of the targets that there exist drugs that can inhibit them, or genetic knockouts. And that's what we used, and found that if we block leukotriene, specifically the cysteine leukotrienes, LTC4, if we block it either by different steps of synthesis, you see on the left here, the avoidance is ablated. And if we use the knockout for LTC4 synthase, it also goes away completely. So the mice now don't distinguish, and they continue consuming, even though the rest of the allergic response is there. They still have intestinal inflammation, they still have diarrhea, they have everything. They just don't understand that what they're doing is wrong. Right? They keep eating something that's bad for them. And so basically what we found is that these two pathways, both initiated by antigen recognition of allergen, triggering mast cells in the intestine, inducing release of leukotriene, LTC4, that acts through two pathways. One, it acts on epithelial cells in the intestine, particularly in large intestine, where it induces GDF15 production. That's been also reported in the context of chemotherapeutic drugs, that colonic epithelium is an important source. How LTC induces that GDF15, we're still working out some details, but it seems like it's definitely downstream of LTC4. The second effect for fast response is through vagus, the population of vagal neurons that are stimulated and go and induce the same circuits that I showed before. So there's this two pathway leading to aversion. And that biology, we think, is a very important part of allergies that we abuse in the modern world, because avoidance, although it's the most effective way and sometimes possible, in many situations it's not possible to completely avoid something, as I described with house dust mite, right? And even with food allergies, where you think, well, avoiding why is peanut allergy is problematic compared to the natural world. In the natural world, you have 150 sources of food, and here we reduced everything to just few, and peanut-based foods become very dominant, hard to avoid. So it's basically the idea that normally induction of these responses, aversion, and all the unpleasant feelings, in addition to expelling something, it induces this avoidance that is a natural and very effective way in the wild. In the modern environment, it's much harder to do that, because we are trapped in certain sort of invariance of the modern environment. But anyway, so this part, what it suggested is that it extends this idea that neurons, it's well known that in the sensory physiology, neurons can sense something directly, like olfactory neurons, or indirectly through the specialized sensory cells, usually epithelial cells, that detect something. For example, taste bud cells detect tastings in the tongue and then communicate to sensory neurons. Cochlear hearer cells detect vibrations, communicate to neurons. Enteroendocrine cells in the gut detect nutrients, communicate to neurons. And what we're adding here is that mast cells, when they detect allergen through IgE, also communicate with neurons. But in that case, what can be detected is now, because it's IgE, it's antibody mediated, it's basically expands the scope of sensory modality almost to infinity, right? Because anything could be an allergen potentially. anything, you can make antibodies to almost anything. So that means that it's a way for the immune system to expand sensory capabilities of the nervous system, of the sensory pathways. And moreover, that expansion can be, for any particular context, would be antigen-specific. So in principle, one can manipulate these types of behaviors at least through specific antigens. And to finish, I want to just put it in the other context of why these findings with avoidance and communication between the immune and nervous system, why they're interesting to us is because it illustrates some principle that is shared in other contexts of associative learning, because this is an example of associative learning. The classical example, of course, is Pavlov's dogs, right? So here we have two types of stimuli, unconditioned stimulus that animal is hardwired to respond to in a very stereotypic way. So meat here induces pre-programmed response, like salivation and so on. Conditioned stimulus, like a ring of a bell, doesn't induce that response. If they're combined, there will be association, and then conditioned stimulus would induce conditioned response. So that's the classical conditioning. In the case of the immune system, we have exact same situation where we have, with a simpler example with the type 1 immunity, we have unconditioned stimulus, which would be microbial structures that are detected by pattern recognition receptors, like LPS, detected by TLR. Pre-programmed response, nothing to learn. It's already all learned through evolution. If we have conditioned stimulus, which would be antigen, which is detected by antigen receptors that are randomly generated, they don't know what that means, so there would be no response. If you combine them, you have association and you have adaptive immune response. And now conditioned stimulus, antigen alone, will induce conditioned response. So it's the same exact principle in both cases. And then there is a phenomenon that makes a connection to the associative learning in allergies. And the phenomenon is what's known as conditioned flavor aversion. This is a phenomenon when you eat something that doesn't agree with you, and then that flavor of that food that made you sick, you remember it and you can never eat it again, right? So very often it comes with seafood, probably norovirus infection or something like that, makes you very bad food poisoning, and then you can't stand the smell of this fried calamari for decades, right? So almost all the people have some experience of that sort. What happens here is that there's some toxin there. We don't have special senses like through smell or taste to detect the toxin. The only way we can detect it is because it made us sick. But we want to avoid it in the future. So what we do, we meaning animals, they will associate it with something that they can detect, in this case flavor, taste and smell. They detect it, make the association, and next time they taste or smell it, they say, okay, I'm not going to eat it because last time I ate it, I was sick. Right? So that's what conditioned flavor aversion. It's one of the most potent paradigms in associative learning because it works with just one try. And so in this case, what we have, something we cannot detect, toxin. We don't have any sensors for it. It just makes us sick. And there's something we can detect, which is flavor. And we associate them. What if there is no flavor? And that's the case with allergic conditioned responses. So again, if you have, let's say, some food antigens and there is nothing noxious, nothing causing malaise, it's just a normal response to food. If there is something noxious there that cannot be detected by any of the systems but would make you sick, you want to associate with something you can detect. If it's flavor, fine, you detect flavor. But if it's not, then what do you do? And that's where the immune system comes in because there's always going to be some protein antigens. It will detect protein antigens and say, okay, whatever made you sick, at the same time in the meal was this antigen. Next time you see this antigen, don't eat it. And that's what would result in this conditioned aversion. That's what I think is the purpose of this response. Both conditioned flavor aversion and antigen-specific aversion, you can think, well, this is kind of silly system because most of the time things will not have toxins. And we know that allergic immunity is error-prone. And that's what I meant in the beginning, that it's not that it doesn't make mistakes. It makes mistakes, but it's very specific types of mistakes. It basically makes false positive errors. And the reason for that is, and that's my last slide, is that every sensory system has to optimize either specificity or sensitivity. There is a trade-off between them. You cannot optimize both. then which one do you optimize? And that depends on the valence of the stimulus. If it's positive valence, if it's something good, like food or whatever, then you maximize specificity, and that's prone to false negative errors. And that basically means that we always want something better, right? If it's negative valence, false negative error would be very costly because that can kill you. It causes illness. What you do instead, you maximize sensitivity, and that is making it prone to false positive error. So it's better safe than sorry. You react to something you didn't have to, but it's better than not reacting when you should have. Right? So that's the idea. So allergic immunity is definitely illustrates that principle, that it's very prone to false positive errors because it maximizes sensitivity over specificity. So I'll stop here. This is the people who did the work. So Stephen Erickson, a very talented postdoc, he did the first part with the help of Jamie and Ben. And Esther and Nate did the work on, again, with the help of Jamie, on the allergen avoidance, with the help of Marcelo's Dietrich's lab, and some reagents we got from these guys. And thank you very much again for your attention.
Well, we do welcome questions. We are past the hour, but this group is conditioned to have lectures until 5 o'clock. So, but probably can't hang out here too long. But maybe handful of questions. Maybe on you were there first. Okay, maybe 4 questions we can do.
Okay. Well, first of all, this was I thought a perfect Bill Paul lecture because of the contributions to type 2 immunity that I discussed here. With regard to the pet shop mice and the hypothesis of repeated or high microbial antigen exposure, I would think that one of the consequences of that would be the development of a much more robust peripheral tolerance system, like the ability to produce IL-10 through either B cells or Tregs and so on. So I was wondering if you blocked IL-10 in that setting, could you reverse this?
Can you remove the brakes on allergic? Yeah. We don't know. We haven't done that experiment. We've done experiments with inducing tolerance in the mice. But, I mean, I see what you mean, and I agree with you completely that they also have, of course, not just increased reactivity to everything, but much increased T-regulatory component as well. And that probably also contributes to it. But we can say that this cross-reactivity covering antigen space by itself is sufficient because we've done experiments with transfers of these antibodies and so on. But I agree that Tregs would be, whether it's IL-10 would be the most relevant, that part I don't know, but this is something we could try with blocking.
Yeah, because in the context of helminth infection in people, that IL-10 has always been like one of the key cytokines.
No, it's definitely something we haven't done specific experiment, but it definitely would be good to try it. Yeah.
Hi. So you made a point that it's the early exposure to antigens that somehow fill out a repertoire, and that's missing in the SPF. But since the antibody repertoire, T-cell repertoire, is almost infinite, what's the hole, and how do you fill it? That is, are there particular specificities, or is it that you're generating broadly cross-reacting antibodies early that are filling out large things, or could one identify specific antigens with some careful work that could plug these holes most optimally and then prevent allergy?
Yeah. So the second part is exactly what we are thinking about trying to exploit, like for therapeutic purposes. But the first part is a harder question. We don't know because what we've tried so far is limited. We're just extrapolating with that model. But I think the notion that we kind of getting converted to is that the idea of antigen specificity may be better, or at least the alternative way to think about it, not specificity, but reactivity. Because specificity is a very relative term, right? And reactivity is more neutral and I think more correct way to talk about it, that you can react to something that may not have been the antigen you specific. But then the question quantitatively becomes how much you cover, how much space you cover. And we hypothesize that you cover, on average, maybe enough, and that what would, on average, provide this state when immune system in the adult is never like truly naive, but there is always some reactivities. But it may be biased to things that may be more common in the environment. That would make sense biologically, but we don't know that. That's the question that's definitely we need to explore further to see whether we can see if there are still gaps in.
Right, things that, I mean, being able to be tolerant to protein-like structures versus some weird chemical structure that would not
right.
be there.
So exactly. So that's the question. One can think of different types of reactivity profiles. So you can imagine something shallow like this, where in the middle there would be the highest affinity, but standard deviation is very high. And these would be some surfaces that may be smoother sort of surface. So you make 10 versus 8 hydrogen bonds, and you find versus hapten, where the peak would be very sharp. And you have to be very, very particular in your structure to detect it. And I think the haptens is basically, I mean, it was a wonderful invention to study immune system, but that maybe biased our thinking about specificity a bit.
Exactly.
Yeah.
Thank you.
Ruslan, fascinating. I also have a question about this early exposure sensitization. So what you described largely fits with the so-called hygiene hypothesis. Yeah. But one variation, I'm not sure if you were saying this, is that it depends on the skewing of the immune system at the time of first exposure. So if you're exposed to a lot of environmental microbes at an early age, that skews you more toward a Th1 type response, and then when you're exposed, you make an IgG response rather than an IgE response, whereas if an infant is more Th2 skewed, then exposure might lead to an allergic sensitivity. And thinking in that way, I wanted to ask you a very practical question, which is, for a long time, pediatricians recommended not exposing infants and young children to peanuts to avoid their developing peanut allergies. But that recommendation has flipped completely, and in the last, 5 or 10 years now, the recommendation is expose your kids very early to peanuts and other potential allergens to avoid their developing these allergies. So how does that fit? And where is that cutoff between sensitization and...
thanks, Jeff. So this is an important distinction. So how much the coverage, antigen coverage contributes versus effector type versus tolerization. So that's what the slide meant to convey, that in the case of this peanut feeding to babies, it's all about inducing this tolerance, expanding Tregs that are specific to peanut antigens. There is that contribution, and in that case, I think protection probably works through that mechanism, as you imply, that it's inducing this tolerance towards the particular antigen. In the case of deviating response to opposite type of response versus antigen coverage, those two is harder to dissociate for us because it is deviated IgG types of antibodies that exactly provide protection. So it's both having those antibodies that cover antigen space and having them of that particular type of IgG type. So those way it's harder to distinguish, and I think they probably don't need, they should be distinguished just out of interest, but I think they generally would operate together. But distinguishing from the pathway that leads to this tolerance through Treg expansion versus antigenic coverage, I think these operate on very different timescales also. This is very discrete sort of events of exposure in early life developing Tregs versus this continuous accumulation of specificities or reactivities towards different antigens that takes maybe years in humans to develop.
Okay, thank you.
Okay. So, well, first, let me repeat the CME code, 57689. We will have a reception, I believe, with peanuts and fried calamari out there, so hurry up. And let's thank our speaker one last time.